6. That is the magic number of marks available here. And the key thing you really need to realize is that these 6 marks are awarded based on a very strict, highly predictable mark scheme checklist. The examiners, they are not looking for creative writing. They aren't asking you to guess some obscure scientific secret. They literally just have a checklist of points, and every time you hit one of those specific points, you get a mark. So your goal isn't to be some brilliant author. Your goal is simply to check their boxes. So, here's our roadmap for this explainer. 1. The 6-mark question. 2. Step 1, identify variables. 3. Step 2, keep it constant. 4. Step 3, method and measurement. 5. Step 4, repeats and safety. And 6, putting it into practice. Starting off with Section 1, the 6-mark question. So, like I mentioned, examiners are grading against a specific list of marking points. They are looking for your ability to select variables, control environments, and measure accurately. And to hit all of these, we're going to use a clear 4-step blueprint. This framework is going to form your core strategy for literally any planning question you will ever encounter on paper 6. Seriously, if they ask about human heart rates during exercise, you use this blueprint. If they throw a totally random question at you about testing vitamin C in fruit juice, or maybe phototropism in seedlings, you use this exact blueprint. Memorize these 4 steps: variables, constants, method, and repeats with safety. Moving right along to Section 2, which is Step 1, identify variables. Reading the question prompt carefully is absolutely critical here to extract exactly what the examiners want you to test. There are 2 core variables that are worth your first 2 marks. First up is the independent variable. This is what you change. For example, if the question asks you to investigate the effect of temperature on amylase enzyme activity, temperature is your independent variable. And here's a pro tip straight from the mark schemes. Don't just write the word temperature. Say, test at least 2 or 3 different temperatures. Okay, then you have the dependent variable. This is the exact result you measure. In that enzyme experiment, you might measure the time it takes for an iodine solution to stay yellow-brown, which indicates starch breakdown. State both of these clearly, and boom, you've already bagged 2 marks. All right, Section 3, which covers Step 2, keep it constant. To make your experiment a fair test, you need to know your magic list of constants. Basically, if you're changing the temperature to see its effect on an enzyme, absolutely everything else must stay exactly the same. Whenever you're dealing with liquids or enzymes, always write that you'll keep the volume of solutions and the concentration of solutions constant. Now, if your independent variable is not temperature, then temperature becomes a constant. And I'm going to give you an absolute cheat code directly from the examiner's mark schemes here. Don't just write keep temperature constant, write use a thermostatically controlled water bath to maintain temperature. I promise you, the examiners love that phrase. Also, if your experiment involves humans, like measuring heart rate after exercise, your constants should be things like the age, sex, and fitness level of the subjects. Write down 2 or 3 of these, and you've scored another mark or 2 effortlessly. Next up, Section 4, which is Step 3, method and measurement. This step is where you briefly write out the actual procedure. But listen carefully, do not write a massive, sprawling essay detailing every single drop of liquid transferred. Keep it simple. To execute your method, examiners are really just looking for standard laboratory practices. So you should definitely mention letting solutions equilibrate to the correct temperature before mixing them. That just means giving them a few minutes in that water bath so they actually reach the target temperature. You should also name specific tools. Doing food tests, say you'll use a spotting tile or a white tile. Measuring a reaction over time, say you'll take samples at set intervals, like maybe every 30 seconds using a stop clock. Specificity with your equipment is exactly what gets you those points. Section 5 brings us to Step 4, repeats and safety. Guys, this step contains literally the easiest bonus marks available on the entire paper, and yet stressed out students completely forget them all the time because they're so intensely focused on the complex biology. So here is a golden rule derived straight from the source material. Always write, repeat the investigation at least 2 more times to find an average or identify anomalous results. That right there is almost always worth a mark. Then, add a safety precaution. Even if you haven't been prompted, write a safety measurement. If you're using chemicals or hot water, say wear safety goggles and gloves. If you're using ethanol for a fat test, write keep away from open flames. Or if you're doing an exercise experiment, simply say ensure the subject is healthy before starting. It is just one single sentence, and it secures your final mark. Finally, Section 6, putting it into practice. Okay, let's look at some actual past paper questions to prove that this exact step-by-step formula works every single time. Imagine an experiment setup designed to investigate yeast respiration using different concentrations of glucose. The student puts a small test tube containing yeast and either a 1% or 4% glucose solution inside a larger test tube, and then inverts it in a warm water bath. After exactly 5 minutes, they measure the height of the yeast suspension. Now, if you were asked to plan this on an exam, how would you do it? Well, let's map it right back to our blueprint to build the perfect 6-mark answer. Step 1, variables. Our independent variable is the different glucose concentrations. We'd specifically state we are using at least 2, like 1% and 4%. Our dependent variable, the thing we measure, is the height of the yeast suspension. Step 2, constants. We absolutely must keep the temperature constant, so we explicitly state we will use a thermostatically controlled water bath. We'd also keep the volume of the yeast suspension constant. Moving to step 3, method. We specify setting a stop clock for exactly 5 minutes before measuring with a ruler. And step 4, repeats and safety. We state we'll repeat the experiment 3 times and calculate a mean. And for safety, we'll keep the setup away from open flames if we're using any heat source. Boom, 6 marks, effortlessly mapped directly to the examiner's mark scheme. Let's try a completely different scenario from another past paper. In this one, a student is investigating the movement of water, which happens to contain a blue dye, up celery stalks at different temperatures. They've got an ice-cold water bath and a warm water bath. They leave the celery in the dye and then cut 5mm sections to see how far the dye actually traveled. Now, even though this is plant biology and not yeast respiration, our 4-step framework still completely dominates the question. Let's break it down again. Step 1, our independent variable is the different temperatures. We'd state we are testing at least cold, room temperature, and warm. The dependent variable is the distance the blue dye moves, which we measure by counting the number of those 5mm sections. Step 2, for constants, we specify keeping the starting length of the celery identical, the actual species of celery the same, and the time left in the dye identical. Step 3, our method includes giving the blue dye time to equilibrate in the water baths before adding the celery, and using a ruler and scalpel to cut precise 5mm sections. And finally, step 4, we write down that we will repeat the whole process twice more, and as a safety precaution, we'll cut away from our fingers and wear goggles. See? Whether it's yeast, celery, amylase pH, or human exercise intensity, the framework easily extracts the exact points you need for full marks. You know, the anxiety around paper 6 is completely natural, but you now have the ultimate cheat code. By simply using this 4-step blueprint, variables, constants, method, and repeats with safety, you transform a terrifying blank page into a really simple checklist. But I want you to ask yourself this before we go. If these fundamental principles of fair testing govern an IGCSE biology exam, how might they also be the exact same principles used by researchers right now in the real world to test the next breakthrough medicine or a new climate change solution? The scientific method never changes. You just know how to fluently speak its language now. Thanks so much for joining me on this explainer, and I'll catch you next time.